To successfully re-engineer complex living systems in the modern lab, we first have to decode exactly how they naturally operate. That is the precise purpose of UBTPC-104, Biosciences. It bridges the foundational biology you learned in high school with the advanced laboratory concepts required for modern biotechnological research. This diagram breaks down the course into two primary objectives. On the left, we focus on the structural blueprints of life, detailing the diversity, organization, and physiological processes of living organisms. On the right side, the focus shifts to analytical tools, familiarizing you with practical biological methods, evolutionary systematics, and an introduction to bioinformatics. By mastering the physical structures of living things and the laboratory techniques used to study them, you acquire the raw materials needed to solve complex real-world problems in agriculture and healthcare. When we examine the vast diversity of the plant and animal kingdoms, we are looking at a massive billion-year-old catalog of genetic solutions. The course maps this catalog systematically. We classify the plant kingdom starting from algae and bryophytes, moving up through pteridophytes and gymnosperms, all the way to complex angiosperms, or flowering plants. Looking at this flowchart, you can trace how a deep understanding of plant structures, specifically roots, stems, and leaves, branches directly into practical applications for crop improvement and plant breeding. We apply a similarly rigorous approach to animal diversity, classifying organisms into non-chordates and chordates by analyzing characteristic features in representative examples like earthworms, frogs, and fish. This animated phylogenetic tree illustrates how we measure the relationships between those groups. It visualizes molecular phylogeny, mapping the evolutionary distance and specific points of divergence between different species. By analyzing underlying DNA sequences, modern taxonomy allows us to trace exactly how these evolutionary connections formed over time. Understanding these complex relationships turns what might initially seem like a static list of species into a predictive map of evolutionary biological capabilities. To utilize that biological map, we have to understand the physical operating mechanisms inside these organisms, exploring key physiological systems, including digestion, respiration, circulation, excretion, and reproduction. These systems are constructed from four primary types of animal tissues: epithelial, connective, muscular, and nervous tissue, each providing highly specific structural functions. This schematic demonstrates why those tissues matter in the lab. It links a model organism, like a mouse, directly to human biomedical pathways, highlighting the shared biology that makes these animals highly effective testing grounds for medical research. Observing these shared pathways requires precise biological methods. We cover the principles of microscopy and staining techniques, which allow us to visually track chemical reactions at the cellular level. These methods let us study foundational physical concepts in real time, phenomena like diffusion across membranes, osmosis, the mechanics of photosynthesis, and specific enzyme activities. Combining an understanding of internal physiological systems with these laboratory methods gives you the technical ability to accurately interpret the results of real-world biological experiments. However, physical lab work is only useful if it is structured carefully. That requires a rigorous approach to experimental design, with strict rules for isolating variables and utilizing proper controls. This animated Punnett square matrix maps out Mendelian inheritance. It provides the foundational mathematical logic required to track how genetic traits are passed down through generations. Because modern biological research generates massive datasets, from inheritance patterns to full genome sequences, we rely on bioinformatics, the computational branch of biology, to manage and interpret that information. Bioinformatics utilizes vast biological databases that digitally map out the entire tree of life, connecting directly back to the phylogenetic concepts and systematic taxonomy we explored earlier. It is the essential analytical tool of the modern lab, translating biological structures into actionable data and giving you predictive power as a researcher. This chart outlines your evaluation scheme. Continuous assessment builds across 3 exams, totaling 25 marks. Below it, the end semester exam adds 50 marks, bringing the total to 75. Notice this passing boundary. You must secure a strict 40